So for you guys on Zoom, I'm just gonna, you can open up the document. Well, I'll open it for you. Oh, yeah, okay. So the last part is basically watching everyone else's videos and then just doing a really brief summary and coming up with a question about it. There we go. Okay. So let me share that with y'all. Okay, finally. Yeah, the disease viewing assignment. The disease video viewing assignment. So this is in that overall kind of assignment document. There's a place for you to upload it. I need to double check and make sure I set it so you can upload stuff to the assignment. But you're basically going to watch everyone else's videos. So there's 8 other groups in here, so you'll watch 7. If you're at the symposium and presenting, if you see someone else's presentation there, you can just kind of take notes on it there, and then you don't have to actually watch the video. But all of the links are on that discussion board still for everyone's video, so you can watch theirs. Oh, there's one that isn't up there, so you only actually have to watch 6. So just watch the ones that are on the discussion board. Obviously put the name of the disease, a really brief summary of it. So really big take-home messages, 5 to 7 sentences, not really all that much. And then tell me one unique thing you learned from the presentation. So something that you found particularly interesting about the disease. And then you have one question. So just come up with one question that maybe something you weren't clear about or something they didn't talk about or something you've heard about that presentation, whatever question you have for them about that disease. And you're going to post, you can write that question in that document with this other stuff that you're uploading to Canvas, or you can just post it on the discussion board. So it has to go on the discussion board either way. I'll look for them there. If you don't put them in this document, that's fine. Kind of up to you how you want to do that. But you'll ask that group, just kind of reply to their post and ask the question on the discussion board. So that's worth 40 points. It's a lot of points, but there's not a whole lot. I mean, most of the time will probably be you watching the video and kind of summarizing it. It is due a week from today, so next Monday by the end of the day. Any questions about that? Pretty straightforward. Just watch people's videos, summarize them, and come up with a question. Okay. Now, so I ended on Friday talking about memory. So the rest of this, I just have to finish up the rest of this nervous tissue chapter today, and then we'll get into some spinal cord stuff. Like I said, the rest of this semester is all nervous system. The nervous system is pretty complex, as I had mentioned. So we'll be done with the tissue chapter, and then there's actually 3 more chapters, which I'll kind of cherry pick information from to talk about on the rest of the nervous system. So we'll talk about spinal cord, and then the brain, and then the autonomic nervous system, which is that kind of unconscious nervous system. Okay. There we go. So memory. Memory is pretty interesting. And how we remember and how we forget is pretty interesting. I'm not going to go into too much detail about this, but I think I introduced this idea on Friday, just that there is a physical basis of memory. So that physical basis of memory is called a memory trace. So basically like tracing that action across synapses in the nervous system. New synapses can be created, existing synapses can be improved. So the more you use your brain, basically the stronger you can make it. You improve those synapses, which basically improves your memory and your ability to learn. Remember this, like I said on Friday, as you go out into the world away from college, we're constantly being forced to learn and continue to learn. Otherwise, you don't use those synapses and you don't use that ability, it goes away pretty quickly, especially as you get older and your brain ages and kind of slows down physiologically on its own. There's two different terms to be familiar with this idea of memory. One is synaptic plasticity. Synaptic plasticity is just the ability of synapses to change. So they can be plastic, so change over time. They can be more effective or less effective, more efficient, less efficient, depending on how much you use them. And then synaptic potentiation is basically the process of making the synapses better, making them more efficient at transmitting information. Over the next few slides, I'm going to talk about these different types of memory. So immediate, short-term, and long-term, pretty briefly, just to kind of give you an overview of sort of how they work. And these three different types of memory sort of correlate with different modes of this potentiation. So how efficient that transmission of signals is across synapses. First, the immediate memory. Immediate memory is the ability to basically hold something in your mind for just a few seconds. So that could be like when you're reading. Hopefully, when you're reading a sentence, you can remember the first part of the sentence by the time you get to the end of the sentence, right? That's actually what we call immediate memory. It may not last much past that. Hopefully it does. Hopefully you kind of remember information for longer than that, but that immediate memory, being able to remember the beginning of a sentence when you get to the end, is the main part of the immediate side of memory. It also provides what we call a sense of the present. So you know you're here now because of your immediate memory. Without your immediate memory, you might think you're in the future or the past. That would be weird. So it's kind of a harder, less tangible sense of the immediate memory than reading, but it does provide us a sense of being here now. Short-term memory can last anywhere from a few seconds to a few hours. So we're talking kind of within one day here with short-term memory. Part of short-term memory is called working memory, which is where your memories allow you to take some kind of action. So if you look up a phone number, hopefully you can remember that phone number for a few seconds until you type it into your phone. It's not really necessary anymore with cell phones, but back in the day when you had to look up phone numbers, being able to remember them would have been the working memory idea. There's sort of two different aspects of short-term memory. One is called tetanic stimulation, and one is post-tetanic potentiation. So tetanic stimulation is going to allow for short-term memory because of the basically rapid arrival of signals over and over again that can foster these brief memories. These signals that kind of keep coming one after the other pretty quickly for short-term memory is going to lead to an increase in calcium ion accumulation in the axon terminal. So the very end of the axon where information is being sent. Remember that calcium, you may or may not remember that calcium, when that enters the neuron, that's what a lot of times releases the neurotransmitters, which allows for transmission between neurons. So with that calcium, you get neurotransmitter release. So with calcium accumulation, you can kind of release that neurotransmitter more frequently and more efficiently. The post-tetanic potentiation occurs where those calcium levels basically stay elevated for a longer period of time, again, allowing for more of that neurotransmitter to be released. So that can actually help remember a memory from a few hours ago. So not just maybe a few seconds ago, but a few hours ago is where post-tetanic potentiation comes into play. Calcium levels just stay up. So all of our memories are basically just a bunch of chemicals and ions being transferred around in our body. It's weird to think about. Okay, so that's short-term memory. So we've got immediate short-term and then long-term memory. There's a couple different categories of long-term memory. One is explicit or declarative. That's where you remember sort of events that you can put into words. So if you went to Disney World when you were a kid and you remember riding some awesome ride and you can talk about it, that would be explicit long-term memory. There's also implicit long-term memory, which is probably, I don't know, maybe more useful, but it helps us remember some basic motor skills, how to walk, how to eat, how to talk, stuff that you wouldn't describe as an event, but they are memories. So at one point you didn't know how to walk. You had to learn how to walk. And from that point on, hopefully for the rest of your life, you remember how to walk. That would be an implicit long-term memory. Improving long-term memory can involve new branching of these axons or dendrites. So basically just increasing the kind of surface area, as you can think of it, as on a neuron. And it can also involve some pretty complex molecular changes, which I'm not going to get into. But you can improve your long-term memory, you can improve your short-term memory, but you have to work at it. It doesn't just happen. You have to, it's like a, I guess, kind of like a muscle. You can think of it that way. If you work it more, then it will be more effective. And then on that same note, how we forget things. It's essentially due to depression of calcium levels. So more calcium is going to lead to more effective remembering. Over time, if a person has depressed calcium levels, say blood calcium levels, that means there's less availability of calcium to the neurons. And that actually leads to degradation of the dendrites. It also keeps the neurotransmitter from being released, right? You need calcium to release a neurotransmitter in a lot of cases. So if there's not enough calcium, that neurotransmitter can't be released, which means that memory is never recalled. And synapses that aren't used are basically removed from neural circuits. And you can think of a neural circuit as like that memory trace. So basically, a memory has to make its way through this neural circuit for you to remember something. If you remove synapses from neural circuits, it makes it a lot more difficult to recall information. So fewer neural circuits is not a good thing. And in a very general sense, that's how we tend to forget things. And now I just want to talk about a couple of different diseases that deal with the nervous system. So I talked about MS. Now I'm going to talk about Alzheimer's and Parkinson's. Have you guys heard of Alzheimer's disease and Parkinson's? I think you, again, have to kind of be living under a rock if you haven't heard of either of these diseases. Alzheimer's disease involves memory loss. Parkinson's is more of a loss of motor function. It's not so much a memory disease. But Alzheimer's disease affects typically older people, older individuals. So they have trouble remembering, they can be moody, and they can also lose some of those implicit long-term memories. So they might not remember how to talk or how to walk or how to eat as the disease progresses. There's still a ton of research going on with Alzheimer's disease. They don't really know the underlying cause. They know some of the symptoms that cause this forgetfulness and memory issues, but they don't know really what causes it. Although I was just listening to a podcast about a recent study that came out that links type 2 diabetes with Alzheimer's. So as if you needed another reason to avoid type 2 diabetes, they've known for a long time that those two correlate. So if you have type 2 diabetes, you're more likely to develop Alzheimer's. And now they're thinking it might be more of a metabolic issue in the brain in dealing with glucose, which is obviously a huge part of type 2 diabetes. But essentially what happens in the brain is that acetylcholine is deficient. So acetylcholine is that really important neurotransmitter that I've mentioned many times. So not enough acetylcholine is going to mean the nerves aren't communicating with each other. And deficiencies in something called nerve growth factor, which is what it sounds like. It helps your nerves to grow and to stay healthy. Individuals who have Alzheimer's, when an autopsy is done on them, some symptoms that help to definitely diagnose Alzheimer's from a physiological standpoint are that the folds in the brain, or the gyri, so all those folds basically atrophy, so they kind of break down. And then there's also these neurofibrillary tangles. So all of the nerve fibers kind of get tangled amongst each other and can't communicate effectively. And then there are plaques that build up. So senile plaques and caused by this protein called beta amyloid. And then the neurofibrillary tangles are kind of the two most obvious symptoms of Alzheimer's when you do an autopsy on a brain. Treatment, there's no cure for it. A lot of the treatments are focused on trying to find ways to clear up that plaque. So get rid of the plaques so the nerve fibers can still send signals effectively. But now, based on that study I just heard about, they're kind of looking into how brain cells use glucose as well and see if they can change it from that more underlying perspective. All right, so Alzheimer's disease, and then Parkinson's disease. Like I said, this is more a loss of motor function. It's not really memory loss, but memory and motor function are both involved in the nervous system is involved in both of those, obviously. Typically, this begins in the 50s, in an individual's 50s or 60s, and there's no recovery, there's no cure. The cause of this is essentially neurons that don't release dopamine anymore. I don't know if I talked about dopamine as one of the neurotransmitters, but it's an important neurotransmitter, and basically it's inhibitory, so it calms down the nervous system. Without that kind of calming neurotransmitter, individuals develop tremors and shaking because their motor neurons are basically overactive. So they're firing too often, so you get those Parkinson tremors. There is treatment. There's no great treatments, and like I said, there's no cure, but they focused on trying to develop a dopamine precursor, so something that will eventually turn into dopamine since the body's not making enough of it. But it is pretty tough on the heart and the liver, so trade-offs there. And then there's also something called an MAOI, which you may have heard on pharmaceutical commercials. They always say, if you're taking an MAOI, don't take this, basically, whatever they're talking about. So that's a monoamine oxidase inhibitor. And it basically just slows down neural degeneration. So it keeps those neurons that aren't releasing dopamine and aren't really active anymore from degenerating. All right. So that wraps up all of the nervous tissue chapter. And right now is just kind of a break in me talking and lecturing. I'm going to have you guys work on essentially sort of a 10-question fill in the blank like I would give you on an exam. So there's a word bank, and for you guys on Zoom, stop sharing. For you guys on Zoom, this is listed, it's on Canvas, and it's called Nervous Tissue Worksheet. So if you want to open that up. and look at it and try to answer those questions. There is a word bank and 10 questions. So try doing it without your notes first. And then if you need your notes to fill in the blanks, that's totally fine. Or you can work with someone else to try to fill those in. And this covers, it's 10 questions that basically cover the entire span of what I just talked about on the nervous tissue chapter. So I'm going to give you guys 2 or 3 minutes, well, 4 or 5 minutes to start with this. And then if you need to go to your notes, that's totally fine. This is just kind of for your benefit. Okay, did anyone get stuck on any of these? Seven. Yeah, so that electrical wave or signal, did anyone get that? What's the electrical wave that gets sent along a plasma membrane? It happened in muscles too. Yeah, action potential, exactly. So 7 is action potential. Definitely an important concept. Anyone on Zoom, did you guys have any that you had a tough time with? Or anyone else in here? Yeah. Yeah, 8, 9, and 10. All right, so 8. Right, so we're at a synapse. We have 2 neurons coming together. Which of those releases the neurotransmitter, the pre or postsynaptic? Pre? Yep, the presynaptic neuron is going to release the neurotransmitter. The postsynaptic neuron is going to bind to it, or it's going to bind to the postsynaptic neuron, and then continue the action potential down. Nine. So I'm talking about an inhibitory GABAergic synapse. You don't even really have to know what a GABAergic synapse is to be able to answer this. The postsynaptic neuron is more or less likely to fire when GABA is released. Any idea? So if it's inhibitory, less, yeah. So an inhibitory synapse, in this case, it's releasing GABA, is going to make the neuron less likely to fire, basically. So it will inhibit the activity of those neurons. That's why it's inhibitory. The opposite would be excitatory. It's more likely to fire. And then 10, yeah, we just went over, that's totally new. Does anyone remember what kind of long-term memory like helps you remember to walk? Implicit? Yeah, implicit. So implicit long-term memory is going to help you remember things that aren't really events in your life. It's just stuff that you inherently know how to do. Explicit long-term memory is, like I said, like an event that you can talk about from years ago. All right, any others that tripped you guys up? What? 3. Number 3. Okay. The visceral motor division. So the peripheral nervous system is that really complicated one that has many, many types or categories and subcategories within it. So you have the sympathetic side of the visceral motor division, which is pretty far down in that concept map that I gave you. Sympathetic and what's the other kind? Parasympathetic, yeah. So the sympathetic and the parasympathetic are the 2 divisions within the visceral motor division. So that's an area, and I mentioned that one in particular. We'll talk about the sympathetic and parasympathetic nervous systems a lot more when we get into the very last chapter that we're going over. But I wanted to include that question mostly because I wanted to remind you guys to look back at how all of that that diagram basically of the types and subtypes within the nervous system. So you have the central and the peripheral. The peripheral breaks down into all these kind of subdivisions. So make sure you know how those break out and sort of what they control. Great. Anything else in there? Yes. 4, yeah. So 4 is a tough one. Was a tough one, at least at 8 a.m. The neurosoma is the neuron cell body. What else comes off of the neuron cell or the neuron cell body, the neurosoma? Yeah, dendrites and axon. Yeah, so the dendrites are usually branched. They're taking in information, and the axon is sending the information away, which was another question. 5. So axon away, just remember that. Okay. Anything else? So that's kind of like a mini study guide, I guess. It's only 10 questions, I know, but those are important concepts for the nervous tissue chapter. So hopefully that was kind of a helpful review at the end of that chapter. Okay. We're just gonna kind of dip our toes into the next chapter here on the spinal cord. Okay, so technically, like I said, we are going into 3 more chapters on the nervous system. So most of the other body systems we talked about, like the skeleton, we did bone tissue and then the skeleton, and that was it. It was 2 chapters, right? The nervous system, as I mentioned, is a bit more complicated. So we've done the nervous tissue, we've done the tissue side, but now there's a lot of different divisions within the nervous system. So we're going to start with the spinal cord, spinal nerves, and then somatic reflexes are basically the reflexes you think of like when the doctor hits your knee and your foot kicks out. And then we're going to go into brain anatomy specifically. And then we're going to talk about the autonomic nervous system, which is the unconscious nervous system or unconscious side of the nervous system. And then that will wrap up, well, the entire semester, but also the nervous system information. I've had to cut this down quite a bit just because of time constraints and the complexity of it. So when you're doing your chapter outlines, you'll probably see a lot more than we go over, kind of per usual. So this first chapter, we're going to do, spinal stuff. Most of you probably understand that the spinal cord is pretty important. You can think of it as the information highway that's sending information up and down your body. So up to your brain and then carrying information from your brain down to the rest of your body. And it has a few really important functions. So I've kind of separated these out to talk about these functions of the spinal cord. First, conduction. It's conducting electrical waves and information up and down the spinal cord, like I just said. This is both sensor and motory information. So information that's being brought in through sensory receptors and then information that's being sent out to the muscles to tell them what to do. It's also a really important center for neural integration. And what does that mean? It basically means decision making. And this is sort of, so your brain does a lot of decision making too, obviously. Neural integration in the spinal cord mostly takes in information that you don't really need to think about. So that's where the reflexes come in. And then takes in information, makes a decision about what to do, and sends out the appropriate motor response. So it integrates all those different aspects of the nervous system. It helps us move. So basically the repetitive sequences for walking. So we do the same things, the same muscles contract over and over in order to help us walk in a very specific pattern. The spinal cord is what allows us to do that. So if you get a spinal cord injury, a lot of times that will affect your ability an individual's ability to walk. And then, like I mentioned, reflexes. So reflexes, we'll talk about these somatic reflexes, which are the typical reflexes. There's also visceral reflexes, which we'll talk about in the autonomic nervous system chapter. But these are just involuntary responses to some kind of stimuli. Your brain doesn't have to think about what to do. Your spinal cord just tells that part of the body what to do. So if your tendon in your knee is getting hit by that little hammer that the doctor has, your spinal cord is basically saying, okay, kick your foot out. There's not really a purpose to it, but there are more important reflexes, like pulling your hand away from something hot, something like that. But your brain doesn't have to think about it. Your spinal cord, the nervous system in your spinal cord takes care of that. Okay, a little bit of anatomy about the spinal cord. It's essentially just a cylinder of nervous tissue. It runs up and down the inside of your spine. So if you remember those vertebral bones, they have that vertebral foramina right in the middle, a big hole. That's where the spinal cord runs through. It's nice and protected inside those vertebral bones. And it enters the brain through the foramen magnum in the skull, that big hole in the bottom of the skull. There's 31 pairs of spinal nerves that come off of the spinal cord. I'll get into a little bit more detail on those later on. But they're essentially named based on what vertebral bone region they come off of. So you have cervical, thoracic, lumbar, and sacral. And then they're numbered. So C1, C2, just like the vertebral bones. You have your C1, your C2, your C3, your T5. Most of the nerves, some of them have like special names because they're particularly important, but they're all numbered based on that same pattern. And there's one coming off of each side of the spine, obviously, because we have two sides to our body. There's a few particularly important regions of the spinal cord. I'm going to talk about them here, and then I'll show you some diagrams. I think it's helpful to see pictures of them. So yeah, like I said, the spinal cord is divided into the cervical, thoracic, and lumbar and sacral regions. There's two areas that are a little bit thicker than the rest of the spinal cord. That's in the cervical region and the lumbar region. So the cervical enlargement and lumbar enlargement are basically thickened parts of the spinal cord to allow for the many nerves that have to innervate our upper limb and our lower limb. And then there's also a region near the base of the spinal cord called the medullary cone. So this is under the lumbar enlargement. And it just tapers down basically into a cone. In that same region, there's something called the cauda equina, which is a bundle of nerve roots. I'll tell you what nerve roots are in a little bit, but essentially a bunch of nerves that occupy the vertebral canal. So where the spinal cord is, they're within that canal from L2, so lumbar 2 to sacral 5, S5. And those are really important in the innervation of the lower limbs. All right, so let's look at what these kind of look like. First, those enlarged areas, the cervical and lumbar enlargements. It's a little bit hard to see here on the screen. They're not massively enlarged. So the spinal cord just expands out a little bit in the cervical region. So it's highlighted orange here. So it expands out a little bit to allow for those kind of bigger nerve attachments that will innervate the upper limbs. And then down here, the lumbar enlargement is highlighted, and it just barely kind of bubbles out a little bit. And you can kind of see the lumbar enlargement here without it being highlighted. It expands. So cervical and lumbar enlargements are important areas of the spinal cord. And then you can actually see where it tapers off here. That's the medullary cone, which I have on the next slide, but it's pretty easy to pick out. So the medullary cone now is highlighted kind of orange. And that's where it tapers below the lumbar enlargement. And you can't see the nerves, obviously, but they're drawn in here in yellow, the cauda equina. Those are those nerve roots, bundle of nerve roots that are running through the vertebral foramina in the lower part of the spine. So the lumbar into the sacral region. Yeah, let's stop there for today. I'll get into the meninges, which are membranes, on Friday. So no class Wednesday, right? You guys are presenting at the symposium. And then no lab this week. So I'll either see you at the symposium or I'll see you on Friday.